Hey guys. Okay, so this is going to be the second of the recorded video responses for the Chapter 19 discussion on Blackboard. This video is going to cover the production of formed elements and the figure that everybody was confused about, as well as the production of red blood cells and hemoglobin and hemoglobin breakdown. So we're going to start with this figure that I got several questions on Blackboard about the production of the formed elements. And what I will tell you is that this figure is not as scary as it looks. The most important thing I can tell you when you look at these figures is look for something that you know. And what you know for sure are these guys down here at the very bottom. These are your formed elements. You know red blood cells. You know the platelets. You know the white blood cells, which is what those 5 guys are down here at this end. Okay? You understand what those are. They are the cells that float around in the blood and they do different jobs. The point of this figure is that all of these different cell types, all of these different formed elements started as a hemocytoblast. And they go through a different series of reactions and conversions to become individual cell types. What drives that difference is a variety of different things, okay? So if this hemocytoblast converts into a lymphoid stem cell, that lymphoid stem cell is then going to differentiate into a lymphoblast, and that lymphoblast is going to differentiate into a lymphocyte. I remind you to think back to AMP1 where we talked about the differences between blasts and cytes, and blasts being those baby cells, the ones that will build things or will start a process, and the cytes being the final product or the end cell of that process. The hemocytoblast can also differentiate into a myeloid stem cell. Now, you need to know hemocytoblast. You need to know the 2 different stem cells. You need to know everybody that is on this level of this flow chart. You do not need to know these guys. You do not need to know these guys here. Okay? All you need to know is what is not crossed out on this slide. Okay? And I'm not going to ask you to fill them out. I'm not going to ask you to list them. But you do need to know that a hemocytoblast that develops into a myeloid stem cell will develop into a proerythroblast if EPO is present, and that once it develops into a proerythroblast, it will eventually become a red blood cell. All the way down here. What is EPO? EPO is a hormone that stimulates the production of red blood cell development. That's erythropoietin. If, on the other hand, that myeloid stem cell develops into a megakaryoblast, using a different compound, a different stimulant to cause that. We're not going to talk about the details of that. That megakaryoblast will develop into a megakaryocyte, and that megakaryocyte is basically a great big cell that will break off little pieces of itself to form the platelets. The other thing that the myeloid stem cell can do is develop into a myeloblast. Now, a myeloblast has a couple of options. It can develop into one of 3 different kinds of white blood cells: the basophil, the eosinophil, and the neutrophil. Now, the thing that determines what a myeloblast will become is a colony stimulating factor. The colony stimulating factors come in a variety of different forms, and depending on which colony stimulating factor is used, you get a different type of white blood cell. That is all of the detail you need to know about that. You just need to know that they help regulate the development of leukocytes. And I'm just going to put WBC here for white blood cell, but you guys know that means leukocyte. Okay? The last option for the myeloid stem cell is for it to differentiate into a monoblast, and monoblasts will eventually grow up to become monocytes, the last of those white blood cells that we talked about. And that is as complicated as this figure needs to be. So make sure that you take the time to really follow the arrows and think about what's happening in this figure rather than just trying to memorize what's happening and in what order. Okay? Hemoglobin. Hemoglobin is the molecule that carries carbon dioxide and oxygen. It doesn't carry them at the same time, but it can carry, or it will carry both, right? Hemoglobin comes in 3 types: the embryonic and the fetal, and the adult, okay? What you need to know here is that embryonic and fetal hemoglobin has a greater attraction for oxygen than adult hemoglobin does. I'm not going to tell you the answer to this question right now. I want you to think about this, and I want you to respond on the discussion board when you think you understand it as to what the answer is to this question that I'm about to ask you. And that question is, why is it important? Why is it important for embryonic and fetal hemoglobin, hemoglobin found in embryos and in fetuses, to have a higher attraction for oxygen than an adult? Some of you have already figured this out. I want the rest of you to think about this question, because this is what I want you doing. I want you moving beyond just memorizing the words in the PowerPoint to thinking about why. Why would it make sense for an embryonic or fetal hemoglobin molecule to have a greater attraction for oxygen than an adult hemoglobin molecule does? Now, I had this question come in earlier about the 3 different forms that hemoglobin can take. And I call them forms of hemoglobin because a single hemoglobin molecule will take on all 3 of these forms throughout its life. It depends on what the hemoglobin molecule is carrying. So a hemoglobin molecule that has picked up oxygen in the lungs and is carrying oxygen is going to be called oxyhemoglobin. When that hemoglobin molecule arrives with the red blood cell at its tissue target, at its tissues, in the system, it's going to release that oxygen, at which point it is going to be a deoxyhemoglobin molecule. Once it forms a deoxyhemoglobin molecule, it is then able to actually hold on to or pick up carbon dioxide, and it will become a carbaminohemoglobin molecule. And then that red blood cell will go back to the lungs through the heart, as we already know, where that carbaminohemoglobin molecule will release the carbon dioxide and pick up more oxygen, becoming an oxyhemoglobin molecule again. Hopefully that makes sense to you. If it doesn't, let me know, and I will see if I can come up with another way to explain this. But you do need to understand that there are 3 types of hemoglobin, and that hemoglobin molecules in general can take on one of these 3 forms at any given point in its life because they carry both oxygen and carbon dioxide through the body. Okay, so we're going to talk a little bit about how we make blood cells, and I just want to reiterate to you the only things you need to know from this list are the stem cells and the proerythroblasts, and that they become red blood cells or erythroblasts. Now, somebody asked me about this figure. I want to run through this really quick. It's pretty straightforward. If the kidneys detect a decrease in oxygen in the blood, that is almost always a function of not having enough hemoglobin to carry enough oxygen molecules. So what that's going to do is it's going to cause the kidneys to secrete a hormone called erythropoietin, or EPO, and that hormone is going to be released to its target cells, which are in the red bone marrow. The red bone marrow is going to use that erythropoietin to increase the production of red blood cells, and those red blood cells, along with their hemoglobin molecules, are going to increase the amount of oxygen that's being carried in the blood. If that still doesn't make sense to you, if you still don't understand that process, feel free to shoot me another message, and I will try to come up with another explanation for you. Now, after 120 days, the red blood cells get destroyed through a process called hemolysis. Hemolysis breaks down the red blood cells and releases the hemoglobin from the cell the macrophages then take up that hemoglobin and will break the hemoglobin molecule down into its subunits. The goal here is to get the parts so that we can make more later. The hemoglobin molecule itself is going to get broken down into two major subunits, the globin chains, which are mostly protein, and the heme groups, which include an iron component and a non-iron component. Okay. The protein is the easiest to understand. The globin chains get broken down into their subunits, which we all know amino acids are the subunits of proteins. So the proteins get broken down into amino acids, and those amino acids can then be used to make other proteins, to make more hemoglobin molecules, or they can be metabolized and used in cellular respiration for energy resource. The iron in the heme molecule is going to get combined with a molecule called transferrin, and that transferrin is going to basically carry or transfer that iron molecule through the blood into a couple of different storage tissues where it can be stored and wait for if we need more, and most importantly to the red bone marrow, where we use that iron to make more hemoglobin in the process of building more red blood cells. The non-iron components are going to go through a series of conversions with the goal of being able to eliminate these waste products from the body. So we're going to start with our non-iron component being converted into a molecule called biliverdin. And biliverdin is going to then be converted into what we call free bilirubin, which I'm going to abbreviate FBR. We're going to take the free bilirubin and we're going to combine it with glucuronic acid, which I'm abbreviating as GA. If you want to know how to spell it, it's in the text on the other side over there. The free bilirubin plus the glucuronic acid will yield conjugated bilirubin. Now, the thing about conjugated bilirubin is that it is water soluble. So when you're looking at these figures, the bilirubin that we have listed here and the conjugated bilirubin that I'm talking about right there is basically the same thing. Okay, that bilirubin is going to get transferred through the blood to the liver, and the liver is going to take most of that bilirubin and secrete it out into the small intestine as part of bile. And we'll talk about what bile's job is when we get to the digestive system. But what happens in the gut is we have bacteria that will break down that bilirubin and will turn it into smaller derivatives, pieces and parts, that then get excreted as part of the feces. There is a tiny amount of conjugated bilirubin that is absorbed into the bloodstream, and those molecules are actually removed from the blood by the kidneys and secreted as urine. So that explains how we break down hemoglobin and what happens to the different pieces. Hopefully that helps you understand the relationships between biliverdin, bilirubin, and conjugated bilirubin and bile. If you have further questions about this, feel free to ask or set up an appointment and schedule a WebEx chat with me where we can sort of talk about this one-on-one and make sure that you understand how this works. That ends the second video for Chapter 19.